across the border, and we have a long-standing cooperation with the North American East Coast research community. And one of the reasons is that we are to some degree upstream in terms of the water masses, so the Gulf of St. Lawrence and some Labrador currents actually flow down south. So there's a tight connection also physically, not only scientifically. And I would like to start with acknowledgement because it's more like a summary of a substantial amount of work which has been done here in the past 6 or 7 years. And results I will present basically part of this finished thesis from Elizabeth Chadwick. And the later part will go or report on ongoing work by William Burt, who's doing his PhD on the lateral carbon transports. And relevant contributions are also by Susan Craig and by our colleagues from the Bedford Institute of Oceanography. And they have an invaluable collection of data sets and expertise in obtaining and interpreting field data. So this is basically the core of the research group. And I might have overlooked some particular names, but in the end I should thank them and tell clearly that they have a major impact on this. So the outline of my presentation, basically I want to focus on the seasonality of the surface water carbon dioxide conditions, which is in the surface water air-sea fluxes, and also surface water pH, which are, at least for this area, the primary controls of the pH and ocean acidification conditions in the upper part of the water column. As a second part, and this is also more a mechanistic approach, I want to speak a little bit what the seasonality regulates, in particular in terms of the biology. And there's a big difference between the winter period, where there's no biology obviously, and then the spring bloom and the post-spring bloom period. And this has, as we see later, a major impact on the conditions relevant for acidification, which would be the saturation state of the carbonate minerals or the pH itself. And I will come back to this in the second half. And in the end, I will spend a couple of minutes on the source of CO2 or on the source of the acidification, where protons or carbon dioxide are delivered to the surface waters and actually regulate on the beyond seasonal timescales surface pH and CO2 conditions. And then I will try to summarize this a little bit from a mechanistic perspective, how biogeochemical systems work in our area. So the Scotian Shelf is basically on the southeastern tip of Canada, or as I said, the northeastern extension of the Nican area. And this plot gives you a rough sketch of the primary hydrographic features. You can see the Gulf of St. Lawrence and the outflow of it through Cabot Strait is basically closing off the Scotian Shelf, and the Nova Scotia current is basically going along the coast from the northeastern into the southwestern direction, and which is more or less a direct outflow of the Gulf of St. Lawrence. And then you can see from the map already that this is the hydrographic connection to the downstream regions in the Gulf of Maine, and potentially even a little bit further down. There's also some impact from the Labrador Shelf current, which makes its flow basically out of the Labrador Sea around Newfoundland, and then goes partially in and out of the Gulf of St. Lawrence, and then goes down southwest a little bit further offshore than the Nova Scotia current. And the orange line is basically a not so nice description of the Gulf Stream, which is the other major water component. So these are the warmer, more saltier waters from the south, and the picture basically shows you nicely why Nova Scotia has very cold winters compared to Europe, because the Gulf Stream takes its turn to the east, just a little bit south of us. So this is basically the main hydrographic properties. And the yellow star on the map indicates where our buoy location is. And this is a time series which has been started late 2006. And it has an autonomous instrument which measures the CO2 and the temperature salinity in hourly intervals, except for maintenance periods. And I will primarily focus my talk on data of this instrument. In addition to this, there's a couple of sections which go off the shore in the southern tip of Nova Scotia here, then along the Halifax line, the Louisbourg line, and also the Cabot Strait line, which are shown again on the next slide. And these are sections which are sampled basically twice a year, and you can see them here on the map. Again, on the map, the red dot is basically the station where the buoy is anchored, and then the lines are these ones, which are part of the Department of Fisheries Oceans monitoring program. It's called Atlantic Zone Monitoring Program, and they go there twice a year in spring, so which is April, and in September, October. And we do about 350 samples each of the cruises. Our intention is de-acidity alkalinity, which is dissolved inorganic carbon, but there will be many hydrographic parameters and nutrients and others, in part biological parameters, determined on these cruises. So for the CO2 relevant investigations, we started, as I said, in 2006 and 2007. And since then, we do whatever is possible to maintain this time series over time. So this is basically my attribution to ocean acidification and the link of PCO2 and pH. In the end, most of the talk will be talking about the pH, because this is in the end the controlling parameter for the pH, because the pH is a little brother of the PCO2. But in the end, they are both tightly connected, and I've given you two examples. One is the theoretical relationship, and one is the actually observed relationship, which justifies the statement in the top left corner that roughly at least the pH is inversely proportional to the PCO2. On the top right corner, I took our buoy PCO2 observations and computed them or compiled them into a pH values, and you can see a nice fit. The R square is about 1. So this is in principle the theoretical relationship which comes from the thermodynamic dependencies in the carbonate systems. The bottom left panel shows you observations where both the pH and the PCO2 have been measured, and this has been shown, given to me by the group in New Hampshire, in particular Joel Salisbury. In the end, you can see the same pattern with exponential dependency. Joe tried to fit through a linear fit, but he could have done also an exponential fit. In the end, you see, of course, that the field observations are more noisy than the theoretical computations, but you can nicely see that, again, there's a very tight and well-defined relationship between the pH and the PCO2. And if you think I talk a little bit too much about PCO2 in the next slides, then maybe keep this conversion in mind and try to translate it into pH. But I will do the translation of particular features which I think are relevant in a couple of slides from now, so that this link will not be lost throughout the talk. If we go through the climatological pattern, which we see in certain parameters, we can nicely see how the system in the Scotian Shelf works. And again, the little map on the bottom right shows you the location where we are in. This is the buoy location, which is in parallel to a weekly to biweekly sampling program by the Department of Fisheries and Oceans. And the top panel shows you the general temperature seasonality observed at the stations. And you can see this basically the temperatures reach near zero temperatures in the winter time, and then in the summer they go up to 18 to 20 degrees. And we see this pattern a little bit clearer on the next slide. But in the end, this is one of the largest temperature amplitudes which can be observed in the world ocean. The water does not freeze, but it's near freezing if the winters are really cold. And then you can see a very sharp and accentuated increase in temperature, and obviously decline in autumn to the winter temperature is the same rapid, because it has to cross a wide range of temperatures. The really most pronounced and accentuated pattern can be seen in the chlorophyll distribution, which is in the end a measure of the spring bloom. And you can see, and this is basically indicated by the red dashed line which goes straight to all panels, that the spring bloom occurs in the late March to early April season. It lasts about 10 days or so, so it's less than a month certainly. It's a very strong signal, but you can see from the concentrations of chlorophyll that basically after the spring bloom, let's say mid-March or end of March, the chlorophyll is nearly indetectable throughout the remainder of the year, and there's no chlorophyll basically produced which can be measured other than, let's say, in the 1 milligram per cubic meter range, or a little bit lower, which is close to the noise. And the consequence of this is well as seen in the two bottom panels. So nitrate or silicate are shown as nutrient examples. Basically, they are rich in winter with concentrations about 5, 6, 7 micromoles each. But as soon as the spring bloom basically occurs end of March, early April, the nutrients are all gone, and the remainder of the year basically the surface waters are nutrient free. This is kind of the biogeochemical background setting, and I will interpret these settings a little bit now in the view of the CO2 and pH system, and try to pinpoint the major processes which drive the surface water conditions on the background of these chemical properties. This is basically what we have as a time series from our buoy location. It shows you the partial pressure of the PCO2 over an annual cycle. And you can see that the data show basically the first deployment in 2007, and then it goes continuously until 2012. And if you see a gap in the data, this is basically when the buoy went out of the water for maintenance, or when it gets run over by a ship and has to be repaired. I think the most striking feature is that there's a very clear and reoccurring pattern in the surface PCO2 distributions. So basically in all years of the observations, we start with winter levels which are on the order of 400 to 450 microatmospheres. And then you can see the very accented drawdown of the PCO2 of 150 to 200 microatmospheres, which occurs in the end of March, early April. And then you can see the recovery occurring in May, June, and maybe a little bit of July, and then flattening out values over the late summer, autumn period, which approached in the winter values. If you see, of course, you can look a little bit more in detail. And for example, if you watch at the drawdown at the spring bloom, here, for example, compared to 2012, which is the olive color and the light blue color, which would be the year 2008, you can clearly see that there is sometimes a couple of weeks difference in the initiation of the spring bloom, and this depends on the local wind and weather conditions, but also on whether we have a very cold or a very warm winter. So in the end, what you can see is that the initiation of the bloom itself might vary over weeks, but in the end, the annual pattern is a very strong reoccurring pattern, with the differences are more on the subordinate level given the major seasonal cycles. The bottom panel again shows you the same recordings for the temperature, and you can see also from the surface water temperature that the features are basically reoccurring every single year, with near zero or below zero temperatures in the winter, and then a rapid increase of the temperature almost about to 20 degrees or so in late August, early September, and then we see a drawdown, a decline of the temperature in the autumn and winter period. And lots of the wiggles which you see in the temperature line are basically from sporadic or high wind events in various amounts of the seasons, which might have the potential to cool down the water column or to slow down the increasing temperature in summer at times. Also interesting is the little blue line on the which is added to the bottom panel. This is basically the mixed layer depth, which has been obtained for 1 year from a Moore profiler. And you can see that the mixed layer depth, or the scale, is on the left-hand y-axis, right-hand y-axis, that in winter the mixed layer is about 60 meters, but as soon as the water basically starts warming, the mixed layer rises very quickly in a couple of days or weeks to about roughly 20 meters, and the excursions basically are caused then by local weather events or other perturbances. If you compile the PCO2 under consideration of the atmospheric CO2 value, which is on the order of 380 to 400 microatmospheres right now, you can come up with an annual flux estimate, which is according to what we have seen before. So we can see that the Scotian Shelf, and this is the negative values, are over the largest part of the year. It's a source of CO2, and actually only the months or the couple of weeks of the spring bloom are able to revert this release of CO2 to the atmosphere and take up for 4 to 6 weeks carbon dioxide from the atmosphere as a consequence, as I said, of the spring bloom. The remainder of the year is basically a constant source function of CO2 to the atmosphere. And if you want, you can establish a mean flux, which is about 0.1 moles per square meter, or the annual flux, if you integrate it, is about 1.5 moles per square meter and year. We did some studies and looked at the interannual variability. So there's water conditions, wind conditions, and weather conditions which alter this. And you can also detect trends on the decadal time series. This is kind of subject to ongoing work, but the broad seasonal pattern is basically depicted here. And what is important in terms of the carbon dioxide and the pH system, of course, if you have outgassing of CO2, you will have a supersaturation, and you also will need a particular low CO2, which basically goes hand in hand with the supersaturation. And the big question, and this will be addressed in the later part of my talk, is where the carbon dioxide and the H+ ions come from and how are they delivered to the surface area of the Scotian Shelf. The biological effects, and this is basically the pH and CO2 control, which is exerted by biology. One way to show them is to look a little bit more in detail into the temperature PCO2 relationship. And the top right panel basically is the data which I showed you in the last slide. Simply I plotted the PCO2 partial pressure on the top right side here over the observed water temperature. And then in the end, what you might do, or what you can do, is you can consider the temperature time scale on the x-axis as a mass time scale. So if you walk from 0 degrees temperature, for example, in the March period, which is the coldest period, then you basically walk the x-axis to the right and increasing water temperature until 20 degrees. So this would be your move in time from spring to the late summer. And obviously in autumn the water cools down, so that your time scale in terms of the temperature scale would be moving the temperature scale from the right hand side back to the cold waters. And the observed PCO2 is shown then in this pattern, so we find basically the numbers back which I have shown you earlier. The winter values in purple, they are the concentrations of 400 to 450 microatmospheres. Then at the coldest water you can see that the spring boom causes a massive drop, which we have discussed earlier, of about 200 or 150 microatmospheres. And then you can see the slow increase with the rise of the water temperature. And then after, let's say, a temperature about 7 degrees or so, which is May or June, the increase of the PCO2 is slowed down a little bit, but still you find the highest numbers in the late summer season. If you want to get rid of the temperature signal, the trick to really unravel the biological signal is to correct the PCO2 data to a certain temperature. This is nothing else than the consideration of the temperature dependence of the Henry constant, and this is basically shown in the bottom left plot. And so the leftover signal, which is actually shown in this plot, is the biological signal which controls the PCO2 and later on also of the pH and the omega, other than the water temperature. And then you see basically what happens over the year. You can see again the drawdown of the PCO2, which is the carbon dioxide uptake by algae in the spring bloom, which lowers the PCO2 from the highest concentrations observed at the lowest temperature to the spring bloom values. And then you see basically a discrimination of two phases in biology, or two further phases. So the first phase in biology is basically the spring bloom lowering the CO2. Then the purple phase, and this is a couple of weeks when the water warms from 0 to, let's say, 6 or 7 degrees, you actually do see an increase of the PCO2 here, and a biologically driven increase. And this means there is no net uptake of carbon dioxide in this period, and this can be attributed to the decay of the bloom. So there's a certain period in time, and we see this later again, where the carbon dioxide is in early spring is basically recovering and not affected by bloom activity, but rather by the opposite. But then we see once the water reaches about 6 to 7 degrees, that again the PCO2 starts dropping again, and the cause for the drop is basically the production of organic matter throughout the late spring and summer season. And the CO2 uptake, so this means the CO2 decline of this line, stops at the maximum water temperature, and then in autumn winter basically you see a recovery of the PCO2 to the high winter values, and this is driven by the respiration of organic matter, and in the later part also by the deepening of the water column and entrainment by outwelling and subsurface waters which are CO2 rich. So what this picture in the end shows it's a seasonal cycle, and the only difference, which I said in the beginning, I didn't plot this over time. I plot this as a function of water column of surface water temperature, and then you can go around basically the seasonal cycle in a loop, and you start in the winter from the high values of 550 ppm, go into the spring via the lowest values and the bloom decay, and then continue to the summer declining CO2 values, and once you reach the summer, basically the respiration takes over the dominant control, and the carbon dioxide system goes back to increasing values. And the consequences of this, I will show 2 slides from now for the pH and also for the omega. But now I want to spend 1 slide more on the biological information which we have to explain this pattern. So on the top left-hand side, you do see again the plot which we have just discussed. And on the right-hand side, this is a little bit a somewhat messy plot. This is basically gives you results of phytoplankton cell counts, and all the phytoplankton groups, they come a little bit with different orders of magnitude, but this is not really the most important thing. The relevant thing is basically if you simply look at the sequence of the peaks, and the plot in detail gives you again as x-axis the time scale. So the left-hand side is the winter value, January, February, and then you go throughout the year until December. And the first peak, basically, this is the gray circles, are the diatom populations, and underlying, which is hard to see, is actually the observed chlorophyll measurements. And you can see that the chlorophyll peak, which is caused by the diatom population, is basically the dominant biological factor which is prominent or existing at this particular season, and this is exactly what you see in the spring bloom. So the argument you can make is, if you compare this to the PCO2 diagram on the left-hand side, that the spring bloom, so the massive drawdown of carbon dioxide during the spring bloom, is basically a function of a diatom growth, and this is also the signal which is visible in the chlorophyll concentrations. If you then remember what we discussed on the last slide in terms of the bloom decay, this was about in May to June. You can see the purple line on the left-hand side slowly increasing, so this is the decay of the bloom. And if you compare this to the dashed line on the right-hand side, you can see that basically this period is exactly between the occurrence of 2 biologically controlled communities. So where the gray line is in about the May on the left-hand side, basically the diatoms have composition have collapsed, but any other composition hasn't had the time to evolve. So what the PCO2 actually shows is very nicely mirrored in the phytoplankton community composition observed over the area, that there's a gap between the spring bloom driven community and the summer community, and in this gap the respiration dominates, and this is exactly what the PCO2 diagram on the left hand shows in terms of increasing PCO2. The second feature which is relevant here is the second peak on the left-hand side, and these are basically phytoplankton structures of smaller communities, so dinoflagellate, pico and dinophytoplankton. These are small groups, and if you remember my initial slide, there's clearly evident that there's no nutrient available, but the PCO2 drawdown still shows you that these communities are active, and in the end the phytoplankton cell count do show you that the smaller phytoplankton groups are the ones which are able to grow, build up biomass, and actually draw down the CO2, and by this regulate the PCO2 and the surface water pH and omega in the late summer and late spring to early summer period. If you also follow the green line on this slide on the right hand side, this is again the chlorophyll concentration, and you can see, and this is a crucial figure if you want to transfer this to satellite observations, that in the end the big bloom and evolution of the smaller phytoplankton group is not followed by the chlorophyll signal, so it's not possible to attribute the phytoplankton groups in the smaller size range really from in situ chlorophyll concentrations. So if you add this all up and make an integration over time, you can say that obviously the spring bloom, of course, is the dominant feature fixing carbon into the biological pool, but the post-spring bloom production, which would be the carbon fixation by the smaller phytoplankton groups, is still on the order of 30 to 50% relative to the carbon fixed by the diatom blooms. And then once you go beyond the end of the summer, which is at 20 degrees, or let's say end of August, you can see the community collapsing of the smaller phytoplankton groups, which in the end means there's a net respiratory system, and your CO2 partial pressure basically recovers by the respiratory activity and goes back to the high winter values. So this was basically the short view on the biological control of the surface CO2 systems on basically two different biological regimes which have massively different carbon uptake rates and obviously exert a massively different control on the pH and the omega, as we see in the slide, where I want to link basically the seasonality of the pH and omega to the observed phytoplankton and CO2 dynamics. And what I did here in the bottom two panels, I took the buoy values from our entire investigation period and computed on a weekly basis annual or average or mean pH and omega values. And you might remember that there are some years where the buoy wasn't in the water, and some years there wasn't. Some seasons are better covered. So this is a pretty broad average. But remember the PCO2 slide, that the signal itself was pretty dominant. So the general patterns are certainly covered by this kind of lump sum integration of the pH and the surface aragonite saturation state values. And what you see if you want to look at this picture in the terms of pH, you can see a certain pH level in the winter time, which is about a little bit more acidic than, let's say, 8. And then, of course, you get this massive increase in the pH by the carbon dioxide uptake, so the reduction of the CO2 concentrations by the spring bloom, which is nicely shown in the difference between the purple and the dark purple and light purple colors indicated by the spring bloom. And then you get a recovery or a decline of the pH as a consequence of the collapse of the bloom, and you can get somewhat moderate values or on a rather homogeneous pH values for the late summer and the autumn season. The omega shows a little bit of a different picture. So the omega is the calcium carbonate saturation state, which is a partially function of the carbonate concentration and salinity or calcium. So you can see that the lowest concentrations, the lowest values for the omega aragonite are observed in winter, which are just a little bit above the value of 1. And then the bloom basically starts increasing the omega, which is a consequence of CO2 uptake, which in turn releases calcium carbonate concentration. And then you can see a further increase in the omega saturation state until the end of summer, which the maximum coincides with the temperature maximum. And you can see then after the temperature maximum, the temperature declines, and basically the omega falls back to the winter values. What you can nicely see from the comparison between the pH and the omega values is the different effects that seasonal warming or temperature has on the CO2 properties. You can see in the warming in the pH basically is outcompeting the ongoing summer production. So if you look at the plot above and look at what we discussed about the production in the late spring and summer period, so there will be continuous CO2 uptake, which in the end should increase the pH on a somewhat weaker or more moderate effect than the bloom, but still it should increase the pH over time. But the temperature, of course, does the opposite. So if you warm the water and do nothing, the pH goes down. And so both effects, the somewhat weaker summertime production and the seasonal warming, basically outcompete each other to some degree, and that's why you see a very or rather homogeneous pattern in the pH in the late summer period. The omega responds differently on the temperature. If you increase the temperature, basically the omega saturation state goes up, and also the omega saturation state goes up by the late spring and summer production. So both the biology and the thermodynamic-driven temperature changes, they pull in the same direction, and this basically is causing the fact that you do see your omega maximum at the end of the warming period, where the water is the warmest and the production basically begins to cease. So this is basically an attempt to really relate what we see in the CO2 and in the biological happening to the seasonal patterns in the pH and the calcium carbonate saturation state. And as I said, these are very broad averages to establish a seasonal cycle, and there's much more details involved, which I don't want to go into right now. So the final question which I want to speak about is the question where the CO2 and also in the end the H plus ions come from, which fuels the CO2 release of the Scotian Shelf. And for this we did a radio tracer experiment, so we measured the radium-224 concentrations along one of the lines which I have shown you initially, and the map where we did this is basically shown in the bottom left corner with the dark red bar. So this is the Halifax line, and remember this, one of the stations, the second one underneath this red bar is the station where the buoy was or is moored. So what we did, we measured the radium concentration in two different depth levels, so in the surface waters and in the 80 meters level. And this is quite a logistic effort, so that's why you don't see the high sampling density which you wish to have, because you need about 200 liters waters per sample. So this is quite some movement of mass on the ship itself. But we did do these two sections inshore, and what the map shows you basically is the radium concentration at the 80 meter levels going inshore. So the right-hand side of the panel is the offshore Atlantic side, and if you go to the left of this panel is basically the inshore side. And I've chosen two ways to describe this. So the upper panel is a simple XY plot of the radium activity against the concentration for the two depth levels, and you can see that the surface is pretty flat and only increases at the left end of the plot, and this is basically indicating that the radium is a sedimentary source. So in the end, the increase on the left hand side in the blue line reveals the terrestrial or the land side source of the radium. The red line is the XY representation of the 80 meter sampling, and you can see a massive increase of the radium activity if you start sampling above a subsurface bank which is about 80 meters deep. You can see a massive increase of the radium which is released from the sediment, and then this is the fact which we exploit here. As you can see, the radium concentrations declining if you move away from the source, so you see a declining concentrations on the inshore direction. And since this is a radioactive mineral, we can derive from the decline in the concentration a diffusion quotient, which in the end allows us to calculate onshore transports of carbon, alkalinity and nutrients which you require to understand the CO2 and pH system. And this actually is shown on this slide, which is sort of the last slide, and I don't want to go too much into the radio tracer characteristics of the radium. I really only wanted to exploit this to arrive at transport estimates where diffusion coefficients are desperately needed. And so this is basically, the three panels show you the same concentration, the concentration of alkalinity, dissolved inorganic carbon and nitrate as nutrient examples for the same sections which I shown you on the last slide, and the boxes and the panels on the left hand side are basically the 80 meter horizon which I've shown you on the previous slide, where we also have the radium and the diffusion measurements. And what you can see basically, if you compare all these parameters, you can see an onshore gradient in the DRC, in the alkalinity, but also in the nitrate from the contour plots. And so if you have an onshore gradient, basically an onshore transport is indicated, and together with the diffusion quotient we are then able to convert the gradient into an onshore transport per time unit. And the gradients itself, they are shown on the bottom panel of the top right corner. So you can see the red squares are basically giving you the alkalinity gradient, which we have seen on the panel C. The blue diamonds, they give you the dissolved inorganic carbon gradient, which are shown in the panel D. And the green, so it's also nitrate, and the green triangles are the DRC gradient, which are shown in the panel D. And the blue diamonds, they are given the nitrate slope. And again, the purple line is basically the concentration of the radium from which we can derive the onshore transport of the three species. What in the end comes out are a couple of fluxes where you can put a certain number on it for mass balance approaches. But the point which is, I think, for the ongoing discussion is more important than the fluxes itself are the ratios of the fluxes, because this in the end, the ratios can identify the surplus CO2 and surplus H+, which are delivered to the shelf. And if you start with the dissolved inorganic carbon or nitrate ratio in this fluxes, the ratio is about 21. And the red feeding ratio, which is a carbon to nitrogen ratio in organic matter, is about 6.6 or 7. And this clearly means that the onshore transport of your chemical properties, they carry 3 times more carbon dioxide than if the biology would be able to fix if all this upwelled nitrogen or onshore transported nitrate would be converted into organic matter. So the ratio clearly shows you that this is a strong CO2 source in excess of what can be fixed by nitrate, and basically the same message is transferred if you plot the DRC over alkalinity ratio. So if you go in surface waters, the ratio is about a little bit lower than 1, which would be a near equilibrium ratio. But you can see that the deep water onshore transport ratios reveals a very strong surplus of carbon dioxide relative to alkalinity, which in the end is nothing else than your CO2 source, or which is nothing else than your proton source. And the proton source basically has been computed in the panel A. So this is the top panel on the right. And if you assume that the equilibrium in the atmosphere would be at 400 ppm of what microatmosphere right now, which is close to what we actually observe, you can compute a certain fraction of carbon dioxide which is delivered to the surface waters beyond this 400 ppm threshold, and also which a certain fraction of H+ ions which are beyond a proton supply, which would be hitting the atmospheric equilibrium concentrations. And doing this experiment, you can basically quantify the onshore transport of excess carbon dioxide, which is basically the one which is released to the atmosphere, but you can also quantify the amount of excess proton, which in the end is the acidification of the ocean by onshore transport of carbon-rich and proton-rich deeper waters. And this is basically what is shown in the top paragraph. And if you take then the diffusion coefficient, of course, you can put numbers in a flux per square meter and year on to this. But mechanistically, basically, this is the source of the CO2 and also the source of the acidification on the Scotian Shelf. So this is basically my summary slide to sum this up. So there's a couple of schematics how you can classify continental shelves or marginal seas, and under this classification, which has been developed by Michel Maybeck, strongly we would think that the Scotian Shelf is either an open platform or an open continental shelf, which is kind of similar. And in addition to this, which is a characteristic feature, and which makes it's a very important point to classify near shore regions, is that the Scotian Shelf is in net an upwelling system, so it can be classified as an upwelling system where deep water is brought up to the surface and then exported out of the region on the surface waters. And this is in the end mirrored in this what we have seen on the last slide. And then you can draw a nice schematic, which we have done on the bottom slide, which brings us back to the biology and the controls of the CO2 air fluxes and the surface PCO2. So in winter you have deep convection, it's cold enough that you have convective mixing, and you bring lots of nitrate and carbon dioxide into the surface waters. You have strong winds, and you have a comparably strong release of CO2 to the atmosphere. And then at the moment of the spring bloom, still in very deep and cold waters, the CO2 basically is taken up by the water column for a couple of weeks. The nitrate and the nutrients are used, and the supply to the surface basically is limited and only will occur on diffusion time scales. And then over the summer period, the water is warming, increasing the surface water pressure, partial pressure of CO2, and lowering the CO2 only a little bit, so you get still a release of CO2 to the atmosphere. The winds are obviously generally weaker than in the stormy winter and autumn period. And once you reach the summer, basically the water cooling leads to a deepening of the water column together with wind-driven and convective processes, and again refueling the surface waters with CO2 and nutrients, so such a system can start all over again at the next winter. So as I said, and this is pointed out in the wording on the top right, there is convective mixing and also episodic upwelling. So on a bulk view or summary view, the Scotian Shelf is an estuarine system which is also an explanation for the CO2 source. On the last slide, I've quantified, or at least pointed to a quantification of the CO2 upward, and also to the related excess protons upward into the surface waters. And we do see, and this is relevant also for our downstream colleagues in the Gulf of Maine, freshwater influence from the Gulf of St. Lawrence, which just will pass the Scotian Shelf and then go down further south. This is what I wanted to say. Thank you very much.